Hello, General Biology, and this is going to be the concluding screencast for Chapter 7, 7172, and we're going to watch this video, and I'm going to narrate the video as we go along. This was done by Harvard, and it is The Inner Life of the Cell. You can YouTube it, and you can listen to the narration. The narration is on a little bit higher level. It's a college level for the descriptions, so that's why I've chosen the unnarrated version to go through so that you can pick up just what you need to know. All right, so let's get started with this. So we're in the bloodstream, so this would be a capillary or a vessel, and these are white blood cells that are moving across. Now these are P-selectin fibers that extend off of the cell wall, or not cell wall, excuse me, plasma membrane, that enable it to move. So they're kind of like Velcro in a way, but the cell is able to move. Here's a version of it where it's not moving so that you can see the connections. So when you see it speeding across, it's just making these temporary connections that enables it to move. All right, I'm going to pause this here for a minute. This is what you're seeing here, the cell membrane. And the various aquas and blues and greens are known as the phosphate heads of the cell membrane lipid bilayer, the phospholipid bilayer. This is a lipid raft, and it's carrying proteins. So these are proteins that you see here, other proteins that are here. The name of the cell membrane, or the model of the cell membrane, is called the fluid mosaic model. And you're going to see why in just a few seconds. Notice how it looks like it actually flows and it moves. It looks fluid. Okay, we called this a phospholipid bilayer because the phospho part are these round circles that are referred to as heads, and they're on the outside of the cell membrane and the inside of the cell membrane. And then these fingerly or hairy type structures that you see here are the lipid tails that are in the inside. It almost kind of makes like a sandwich. And this is a phospholipid bilayer, and it's selectively permeable. And we're going to be talking about this in section 7.3 in more detail. But it's selectively permeable, and it allows materials to pass through. These are different proteins, and there's various different proteins that we're going to see. There's going to be structural proteins that are part of the structure of the cell membrane. There's going to be transport proteins that will help materials move into and out of the cell membrane, and then there's other proteins that are going to help with reactions along the cell membrane. Okay, so here's a receptor protein that's going to help receive materials that need to come into the cell. A lipid raft floating by. Okay, now we're inside of the cell and we're looking up at the cell membrane. And here is a transport protein that is embedded all the way through. Another protein that is used for other purposes, not necessarily structural. These are part of your microtubules. These are special proteins that help to form part of that cytoskeleton that we talked about. So we're inside the cell and we're looking up at the cell membrane. Okay, now we're descending, and these are microfilaments, and they're made out of actin. And you can see there's different size filaments. And remember, they're fine and they're flexible. They can be easily assembled and disassembled, as we're going to see in a moment. These are the filaments coming together, the actin coming together to make a filament. So as the cell's forming, that's how they can easily be formed. And okay, so the severing protein is going to come in. Those are the actin. And here's that severing protein, and that's going to come in and break the microfilament apart so that materials can move through, so the cell can go through its regular processes. This is the formation of a microtubule, and these are tubulins. And so you can see how easily a microtubule can be assembled, and then it can be just as easily disassembled. So during the different processes of the cell cycle, it's needed to build up microtubules and to break down microtubules so that the cell can divide. Here's that wonderful, I'm going to pause this here a minute, that wonderful motor protein that is a specialized protein that's using this microtubule to transport the vesicle. The vesicle could have come from the endoplasmic reticulum, or it could have come from the Golgi apparatus, and can be carrying undigested materials, digested materials, special enzymes that need to be exported, special proteins. A number of different things can be in this vesicle. The motor protein, this is like one of my favorite things to see in the cell. And this is happening inside every one of your cells right now. There's a mitochondria. It's using microfilaments in order to move. Okay, I'm going to stop this so that we can orient ourselves a little bit. The centrosome is the area where centrioles come together, and these are microtubules and microfilaments that are coming off of the centrioles, and these two are centrioles, so this is an animal cell. We have a mitochondria that's present here. This looks like it's the Golgi apparatus in the background, and this could be, I'm not sure of the angle here, but this could be a vesicle, but behind you is the nucleus. More microfilaments, as you can see. Okay, so now this is the nuclear membrane, and these are the nuclear pores, and RNA strands that have just copied the DNA sequence are coming out. And you'll see ribosomal subunits that will attach to these ribosome RNA chains, and they're going to copy that sequence, and these are polypeptides, parts of proteins that are being produced. So here's more proteins being produced, 2 ribosomal subunits, another mitochondria. Okay, more subunits. This is a pore on the endoplasmic reticulum, and when the ribosome attaches, it can put the polypeptide chain right into the endoplasmic reticulum so it can be transported. Here's a closer look. And in the ER, these polypeptides will be chemically modified and transported. When modification is finished, vesicles will break off of the endoplasmic reticulum, the same types of vesicles that this motor protein is pulling along. And the membrane structure of this is the same membrane structure as the plasma membrane, so it's going to be able to bond and fuse with that. Here's the Golgi apparatus, stacks of flattened sacs arranged 3 to 7 sacs per stack. It looks kind of like a lava lamp, and these are the vesicles that are fusing with the Golgi apparatus to continue the sorting, the packaging, and the chemical modification of proteins. Here's this vesicle. This is what's inside of it's going to be removed for export, so it will fuse with the cell membrane. So it fused with it, and what it was carrying now is released. And this is how materials inside of the cell get transported or exported out of the cell. So again, we have our special protein that's going to be able to receive things that need to come into the cell. Another transport protein. These always remind me of transformers. See the raft? Again, they're specialized proteins. Integrin is a type of protein. And they're going to help with the movement and communication of that cell with another cell. So there's that white blood cell again. And it's flattening itself and going into the bone marrow, into a bone cell. So how cool is that? So that's the cellular activity that is going on in your cells and every single one of your cells that have a nucleus right now. And that's what we've been talking about, all the functions that we've mentioned. All right, so this ends section 7.1 and 7.2. You're going to have a test on this. Have a good one.